Hello, this is an introduction into data analysis and visualizations of the cancer genomics datasets from TCGA using SEMA. So, in SEMA, the basic principle is in asking the questions and forming hypotheses as graphs, where nodes represent your biological or clinical variables of interest, and links between them represent the purported relationships between them, or basically the hypothesis that you're interested in testing. So here, this is the graph area where you make your hypotheses, and this is the plot area where you'll be visualizing the data that is in the graph window. So you start first by populating your graph area with the biological variables or clinical variables of interest to you. So TCGA has a bunch of data types. So you have mRNA expression data for gene-based mRNA expression data from RNA-seq data, microRNA expression data from microRNA-seq data, protein expression data from reverse phase protein array data, somatic mutations from whole exome sequencing data, germline variations from whole exome sequencing data of normal samples, somatic copy number variation data from SNP6 arrays, some of the pre-computed tumor features that we will talk about later, hopefully in a later tutorial from the PanCan consortium, and clinical variables. It also allows you a factor analysis, but we will get to factor analysis later. So right now, let's start by analyzing mutation frequencies in a gene of interest. Let's say p53. We just select somatic mutation, and we type in the name of the gene that we're interested in. So in this case, it's p53, and just say add. When you add that, now you have the node that represents the variable of interest. In this case, it's somatic mutations p53. So when you click on it, the distribution of the data, basically the mutation frequencies, will now appear in this window. So right now it's showing breast cancer. You can analyze for bladder, there's a cervical, colon, or you can look at them all together or just some of them. So it's going to show you the ones that that are selected. So you can either look at them at the whole numbers here. So the red represents mutations, gray represents wild type, or you can look at the proportion. So now it's showing the percentage basically. So in bladder it's almost half and half. In colon, most of them have mutations in p53, and so on and so forth. So next, let's analyze, let's say, expression levels of a p53 target gene. Let's say MDM2. Click add, and now we have MDM2 RNA. And notice that the two have different shapes. So every data type has a different shape in this graph. You click on it, and now you're going to see the distribution of expression of MDM2 across the different cancers. You can log scale it to make it better. Overlay data points or not. Let's actually print it this way. And look at them individually and so on and so forth. Now, let's ask the question, do p53 mutations affect MDM2 mRNA levels in cancers? So for that, all we have to do is draw a link from p53 mutation node to the MDM2 RNA node. And to draw a link, we go to p53 mutation node, click on it, and drag a line to the MDM2 RNA. It's gonna the line is gonna snap. And now we have our hypothesis. So our graph now has a hypothesis. The hypothesis is that p53 mutations affect MDM2 RNA levels, which can be easily tested. First, let's just visualize the pairwise distribution. To do that, you just have to click on the link. Now click on the link, and now you'll see the expression of MDM2 mRNA stratified by mutations in p53. This is for esophageal carcinoma, this is for glioblastoma, or you can look at all of them together. It's too messy, so yeah, just a few of them, a few at a time is also possible. Now, to get statistics on this, you just click on Evaluate, and now the association of p53 mutations with MDM2 RNA levels are evaluated across all cancers, and the statistics are overlaid on top of the links. So the color of the link shows the direction of impact. So blue means negative. So whenever there's p53 mutations, so the MDM2 RNA is reduced, which is what you expect, because p53 is a positive regulator of MDM2 RNA levels, and the number is basically showing this. So it's you can show the z-score, or you can show the p-value of correlation, or this is just a beta value from the regression analysis. I like z-scores because it shows both statistics and the direction of impact. The coloring here represents average across all the data sets, but you can also visualize individual, like for breast cancer, this is way more significant. If you look at the p-value, this is a pretty significant p-value for breast cancer. So when you so after evaluating, if you click on the link, now there is another option appearing in the plot area, where you have the option of either looking at the raw data as you did before for this link, or you can visualize the statistics after being evaluated. Now let's analyze z-scores across all cancers. It's showing you the statistic, the t-statistic of correlation of p53 mutations with MDM2 RNA levels. So as you see, it's mostly negative across all cancers. You can visualize the p-values. This is negative log 10 values, and you can see that these are pretty significant. So basically, p53 mutations negatively affect MDM2 RNA levels in almost every cancer we see here. And you can complicate this this plot, this model further. Now let's look how MDM2, because MDM2 is frequently amplified in cancer. So let's look how that factors into MDM2 expression. Let's select copy number variation, type in MDM2, add, click on MDM2 copy number, and now you see the distribution. This is log scale, so this is not supposed to be log scale. And here you have its distribution across all the cancers. So let's see how MDM2 copy numbers factor into MDM2 RNA levels. Click. Of course, this is showing now across all cancers, and yeah, the scatter plot, and let's see, log scale, and there's a pretty good linear relationship as MDM2 copy numbers increase. Copy number variation increases as so the MDM2 RNA levels also increase. So you have a very good. This is basically a subpopulation where MDM2 is amplified. We can also look at individuals in bladder. Yeah, pretty obvious amplification of MDM2 in many different cancers. In GBM, there is a clear subpopulation that have high-level amplification of MDM2. Now let's analyze them together. So obviously, this you can see from the thick red line that the MDM2 copy numbers significantly contribute to MDM2 RNA levels, as you also see in the individual data plots. So with the z-scores, and yeah, this is kind of obvious. So let's see if the contribution of p53 changed after factoring in MDM2. So it's not as strong as it used to be before we added MDM2 copy numbers, because now the individual statistics are going to change, obviously, because as you add more variables, this is going to be now a multivariate analysis. So p53 what you see here is the partial contribution of p53 mutations having control for MDM2 copy numbers, and that's basically what you see here. But still, we see that p53 mutations significantly contribute to MDM2 RNA levels, even when you factor in MDM2 copy numbers. And again, you can complicate this. Let's add clinical data. There's overall survival, which is the obvious one: age, gender, race, tumor stage, menopause status, vital status. Let's add just overall survival. Oh. Now, when you click on overall survival, you're gonna see Kaplan-Meier curves, and right now we're just looking at it alone. So all you see is just the Kaplan-Meier curves for each cancer. But now let's see how, let's say, p53 mutations affect overall survival. Click on the link, and now in the selected cancers, this is the Kaplan-Meier curves of p53 mutant versus wild type, and mutants are doing worse. We can look individually. Let's add this to this mix. Well, actually, let's also add MDM2 RNA level. So let's see how each of these contributes to overall survival, having control for each other. So basically, we're going to be looking at the partial contribution of each of these to overall survival in different cancers. So evaluate. So from the average, it looked like MDM2 copy numbers have the most significant effect, but let's just analyze them individually. So this is z-scores. Let's look at p-values. This is right around like 0.05 p-value, and this is quite significant. The thyroid carcinoma. Let's look at MDM2 RNA levels. In esophageal carcinoma, it's pretty significant contribution. And p53 mutations probably have a more widespread effect. Now let's analyze head and neck cancers here, where p53 mutations have a significant effect. Let's look at the raw data values, and there you go. So mutants are doing significantly worse in head and neck, and again, the p-value for that association is right here. Let's look at how copy numbers affected MDM2 copy numbers affected the expressions in thyroid carcinomas. Let's go to thyroid carcinomas and analyze individual. We can again you can adjust the cutoff for high and low here, and see how that affects. So high copy numbers are generally doing worse. High MDM2 amplifications. All right. Okay, so that was basically it. So this was a simple introduction to making hypotheses and analyzing them in SEMA. In next tutorials, I will be adding. I'll be going through other features in SEMA, such as factor analysis and more complicated hypothesis testing.